Our first speaker is going to set kind of the baseline for us this morning. Dr. Chakrabartty from the University of Toronto is going to actually be speaking about the ABCs of TTR.
Okay, thank you very much. There we go.
There you go.
There we go. Okay, great. Yeah, thank you, Meryl, for this opportunity. It's great to be here. Great to see all of you people, and great to talk about this subject. My name's Avi Chakrabartty, and from the University of Toronto. And today I'm going to talk to you about molecules that cause ATTR and molecules that treat ATTR. Let's see if this works. Okay, there we go. So these are my disclosures. Okay, so let's get started. Okay, so from the start, I want to emphasize that ATTR it's a molecular disease, and it's unlike cancer. Cancer is not a molecular disease. Cancer is a cellular disease. In cancer, you have cells that behave badly and cause disease. With ATTR, you have molecules that behave badly and cause disease, specifically this molecule here. So this is transthyretin. And before I talk about the disease mechanism and treatments, I'm going to have to give you a little protein structure tutorial. So let's start that now. So this is what we call an all-atom representation of TTR. And so here, every atom is represented by a sphere. Carbons are gray spheres, oxygen's red, nitrogen's blue, and you can see 2 yellow sulfur atoms in there as well. And so this is, the all-atom model basically tells you what the shape of the protein looks like. And to me, it kind of reminds me of a roast chicken. You can see the 2 drumsticks down below and the wings wrapped over the breast, and there's the neck on top. But here's another model that you might remember from high school. It's the ball and stick model, right? And the problem with this model is there's too many atoms. You can't see the forest for the trees. So usually when we look at protein structures, we look at a part of the protein or a subset of the protein atoms at a time. So for example, if we were to zoom in right into the core of the protein, what we would see is the protein is made up of one long chain, which I call a main chain. And I've shown that main chain in green. And so this main chain meanders around and makes up the entire protein. And then sprouting from the main chain are small side chains. And here, what I've done is I've taken the transthyretin molecule and I've deleted all of the side chains. And so this is just the main chain. It's called the main chain model. It's basically like the skeleton of the transthyretin model. And so this is the skeleton of transthyretin model when it's in its normal, natural, native state here. Okay, but if you look closely, you can see that it's made up of sort of repeating structures. There's like short strands, a couple of strands up at an angle up there, some that are horizontal, and you can see a couple of strands connected by loops, right? So what turns out that protein structures are made up of 3 types of substructures. There's the strand, the loop, and the helix. And what we have is we have computer programs that highlight these substructures from the main chain model. So this is how that works. So you plug in the main chain model, and the computer spits out this ribbon model. And the ribbon model basically shows you where these substructures are, where you have the strands, the loops, and the helices. Okay, so let's have a look at TTR a bit more carefully here. So this is TTR. Now with this representation, you can see there's a remarkable amount of symmetry here. there's a vertical axis of symmetry, a horizontal axis of symmetry. And here I've compared TTR to a number of other proteins. So on top is the small protein insulin, made up of only 2 strands and 6 helices. Underneath that is hemoglobin. It's made up of only helices. It has no strands. And then beside that is the Y-shaped antibody molecule. And that's got a nice assortment of strands and a couple helices, not unlike TTR. Okay, so that's the end of the protein structure tutorial. And now we can actually talk about transthyretin and ATTR amyloidosis. And right, so now I want you to concentrate on the molecule on the left. That is the ribbon diagram of transthyretin when it's in its well-behaved state. And you can see I've colored it 4 different colors. And that's because it's made up of 4 different chains: a red chain, a blue chain, a yellow chain, and a green chain. So each of these chains fold up into a structure which is called a monomer, and 4 of these monomers come together to form this well-behaved TTR molecule. And when it's in this state, it carries out its normal function of transporting thyroid hormone and vitamin A. But as we'll see, under certain circumstances, it can start to behave badly and lead to the formation of these other structures which impact in the heart and cause problems. Okay, so fortunately, we do have treatments. And all of the treatments we have target something called the central dogma of molecular biology. So what is the central dogma of molecular biology? Yeah, well, it starts with the famous molecule on the left. That's DNA. That's a ribbon diagram of DNA. It's a double-stranded helix. In between the 2 strands are the base pairs. If you look really carefully, you can read the sequence A, C, T, G, C, etc. And this piece of DNA is part of the gene for transthyretin. And so the gene for transthyretin produces, through a process of transcription, produces the messenger RNA for transthyretin. And then through a process of translation, the messenger RNA is used to make the protein for transthyretin. But we're not done yet. To be transthyretin, you have to make 4 copies of this protein, and all 4 of them have to come together to form the tetramer. Okay, so how do these treatments work, and how do they? So the first treatment, CRISPR-Cas9, targets the first molecule of this long pathway, which is the DNA molecule. So CRISPR-Cas9 knocks out genes. And so the idea is if you knock out the gene for transthyretin, then you won't make any messenger RNA for transthyretin, and that won't, which can't be translated to protein, and you won't have any TTR to form amyloid. Okay, so this is a remarkable achievement. It's the first in-human gene editing therapeutic ever made. The phase 1 trial was quite successful. Now, because this treatment does knock out the normal function of transthyretin, you have to take vitamin A supplements. Okay, so if anybody asks you, CRISPR finds the gene and Cas9 inactivates the gene. Okay, so we can also target the RNA. And so this method is similar, somewhat similar in principle to the CRISPR-Cas9, except we are degrading the messenger RNA this time. So the way it works, you degrade the messenger RNA for your TTR, you can't make the protein, you have no protein to form amyloid and cause disease. So there's different ways of degrading the RNA using, one method is small interfering RNAs, and the other is antisense oligonucleotides. Small interfering RNAs are unique double-stranded RNA molecules, and antisense oligonucleotides are single-stranded. And also, because this treatment does knock out function, you need to take vitamin A supplements. Okay, now we can talk about protein targeting. But before talking about protein targeting, we have to discuss in detail TTR behaving badly or misfolding. How does this happen? Well, we'll start over on the left with the native tetramer. So that's well-behaved native tetramer. That's what TTR is doing its job. That's what it looks like. And then either as a result of mutations or environmental toxins, there's an instability in the tetramer and it falls apart into monomers. So you get all of those monomers forming. And the monomers by themselves aren't all that stable. The monomers, they begin to fluctuate and they don't have a structure and they're flopping around like this. And whenever they form like a flat monomer, that flat monomer tends to stick with another flat monomer. And whenever these flat monomers form, you're getting this stacking up. And that continues, and then eventually you get the amyloid fibril. So that's, and it's this amyloid fibril that impacts your heart, and you can see it right around your muscle fibers. Here, it's all, the amyloid is stained dark red or dark brown. And you can see it's around the muscle fibers. And this is with a light microscope, regular light microscope. And now if you look at that with the electron microscope, you can see the fibrils themselves. They kind of look like angel hair pasta, you know. And then with a fancy new cryo-electron microscope, you can go look much deeper and you can see the actual protein substructures like the loops and the strands. Okay, so how does this protein targeting work? Well, one method is the TTR tetramer stabilization strategy. And this relies on this unique molecule called tafamidis. So tafamidis is basically a superglue. So if you look, tafamidis, 2 molecules of tafamidis, goes and it slips in right at that interface where all 4 monomers are touching each other. And so this is some super glue. You put a dab of it on one side, dab on the other, and this cements the tetramer together. And when you have a hyperstable tetramer, then it doesn't fall apart into monomers, and you don't get any amyloid forming. And so, yeah, we have a couple of different formulations of tafamidis out there that are approved, and next generation molecules are being sought after as well. Okay, so we can also talk about antibody therapy for ATTR. So how would this work? So you probably all know that antibodies work by binding onto things, right? So if a bacteria enters your bloodstream, antibodies in your blood recognize the bacteria and bind onto it, essentially tagging the bacteria with the antibody. And then what we also have is white blood cells in our blood called macrophages. And these macrophages like to eat anything that's tagged with an antibody. Now, applying that to this case over here, if we could tag all of the misfolded states, which are in the red box over there, with an antibody, then we could get the macrophages to eat all of those states. And we have to make sure that those antibodies don't bind the native tetramer, the well-behaved tetramer, which is up in the green box over there. So how do we do this? Well, by looking at protein structures. So here I've shown, so I have this dashed line. Molecules to the right of the dashed line are molecules that are well-behaved, are not well-behaved, that are badly behaved. Okay, molecules to the left of the dashed line are the molecules that are well-behaved. You have ribbon diagrams on top and all atom representations on the bottom. And so just concentrate on the monomer for a second. Look at the ribbon diagram of the monomer, and you see that red strand. So that red strand, it's nice and exposed in the monomer, like in the, and it's also exposed in the misfolded monomer when it's flopping around. And even in the amyloid fibril, that little red bit is all exposed along one side of the amyloid fibril. But now if you look on the other side of the screen at native, natural, well-behaved TTR, you see that that red bit is buried. So now if we make an antibody to the red bid, that antibody will be able to bind the monomer, the misfolded monomer, and the amyloid fibril, but it'll leave the native, natural tetramer, the well-behaved TTR, alone. Right, and so this antibody is the PRX004, which is in the phase 2 clinical trial right now. Okay, so that's essentially my talk. So I hope I've shown you that ATTR is the result of a long, complicated pathway, but we do have treatments that target just about every step of the pathway. And so these are the internet resources I use to make all the pictures, and these are my acknowledgements. Yeah, thank you.
Thank you, that's really nice.